All right, guys, we're going to start here on Chapter 6. I'm going to break Chapter 6 into two video lectures. The first one here is going to be about some basics, some generalities of cells and different types of cells. Break down the difference a little bit here between eukaryotic and prokaryotic cells. And kind of briefly discuss some of the things that you probably already know a little bit about, like the nucleus and ribosomes and things of that nature. In fact, you guys probably know quite a bit already about a lot of these organelles. This is stuff that you guys kind of, I would imagine, have learned ever since you started, life sciences. There are a few of the different organelles that I'm going to go a little bit deeper into detail about, and those will be addressed further in the second video, which deals more with the endomembrane system, is what it's called. So, without further ado, I'll go ahead and give you the brief introduction here about the tour of the cell or the cell organelles, give you some of the simpler stuff, and then I will put together the second video going into a little bit more detail of some of the things that are kind of, I'm not going to say looked down upon, but almost sometimes forgotten when we talk about cell organelles. Okay, so let's just jump right in here. There we go. Okay, that's just a pretty picture of a cell. In fact, one of the things that you see in this picture here is a fluorescently dyed cell. And this cell has different parts of its structures dyed with fluorescent dyes and then lit up by lasers. And the components that you see here most prominently dyed are things called the cytoskeleton. We're going to briefly touch on the basics of the cytoskeleton in the second lecture, but that's not one of the things the College Board really needs you to understand about, so we'll kind of brush it off at least a little bit. But I always include this because I think it's a really cool picture and it provides a different look into cellular structure than what we normally get. Okay, so let's just start here with section 6.2 in the book, which outlines the differences between eukaryotic cells and prokaryotic cells. There are two basic different types of cells that we generally kind of categorize. One is called the prokaryote, one is called the eukaryote. The one on the left in this picture here you see is, get my pointer out, that this guy right down here is called a prokaryotic cell, and for the most part they are simple, super simple versions of eukaryotic cells, which we see over here to the right. Okay, you can see that the complexity of the eukaryotic cell is far greater than the complexity of the prokaryotic cell, and there are quite a few differences between the two, in addition to the complexity that I mentioned there. So we'll touch on some of those as well. Incidentally, the prokaryotic cells are commonly called bacteria. Okay. All right, so before we start kind of breaking down the difference between these two, and we're not going to get real deep into the differences between prokaryotic and eukaryotic cells, I want to just touch on the basic features of all cells, both prokaryotic and eukaryotic alike. Both prokaryotes and eukaryotes both have plasma membranes, and we're gonna go deep into plasma membranes on Chapter 7. So that's about all I'm going to touch on, as the fact that these two types of cells both have plasma membranes. Both of them have a semi-fluid substance called cytosol. Okay, you guys commonly know this as cytoplasm. The book refers to it oftentimes as cytosol. Either one is fine. I will use them interchangeably, and it is just that. It is a semi-fluid. When we think about cytoplasm or cytosol, one thing we have to think about is the fact that true cytoplasm, true cytosol, is a lot thicker than we give it credit for. I've gone to a few workshops at U of I in different labs, and that's one thing that they always kind of harp on, the difference between what we think about cytoplasm and what is actually true about cytoplasm, is that cytoplasm is actually a lot thicker than we actually realize it is. Okay, it's almost a more Jello consistency, and if you think about what Jello is water-based. It's just very, very thick. Okay, both types of cells have chromosomes or DNA that carry the genes. We'll talk about a little bit the difference between where those chromosomes are kept in prokaryotes versus eukaryotes, but they both kind of do the same thing. They carry the instructions for those particular cells, and both have ribosomes, which make proteins for the cell. Okay, so those are the commonalities between both prokaryotes and eukaryotes. Now, let's just dive real briefly into the characteristics of the general prokaryote. Okay. Prokaryote cells have no nucleus. That's the big one. And the reason that they have no nucleus kind of brings us all the way down to, let me see if I can, this is my new pen feature. I got to take a picture of this thing. There we go. That's weird. The reason that prokaryotes have no nucleus is also linked to this right down here, that prokaryotic cells lack membrane-bound organelles. And if you look at what the nucleus is inherently, it is a membrane-bound organelle. So you know, if you're looking at a microscope, that if you don't see any straight up organelles like we normally would see, that you're looking at a prokaryotic cell. In fact, with a lot of these things, you will see something called a nucleoid region. Okay, the nucleoid region is where the DNA happens to be in a prokaryote, so it might look a little bit darker because the DNA is probably stained, but it is not a nucleus in the sense that we think about the nucleus of a eukaryotic cell. Okay, and so the fact that prokaryotes have no membrane-bound organelles means that their metabolic functions are not compartmentalized. Terrible handwriting, but that's what I get for trying to do it on a computer screen. The metabolic functions are not compartmentalized. We see a lot of the metabolic functions in our cells put into different areas because they're needed. The product of that metabolic function or that chemical reaction might be needed in that specific area. Now, with prokaryotes, they do something, again, similar, okay, but it's not bound by these membrane walls, these membranous walls. The prokaryotic cells actually will have different regions that these different chemical functions might happen, but they are not bound up in these little containers like ours do. And so that's a nice option for eukaryotic cells to have, and it's one of the reasons that we've increased in our complexity as well. It allows us to do, or it allows our cells to break down certain types of chemicals in very specific areas and then ship off the products to very specific places or use the products in very specific places. So that allows some variability in construction, and it allows some versatility and variability in the diversity and the complexity of our cells as a result. So generally speaking, prokaryotes are really quite simple. They're pretty simple little things. This is a picture of a prokaryote, and most of the differences that you can see here come with the kind of the outer areas of this bacteria, this prokaryote. Oftentimes, bacteria will have a cell wall, okay, and that's not necessarily something that we've talked about at length yet. Let me get this again. But the cell wall right here is this orange layer, okay, right here. And so that orange layer is not present on every bacteria, but it is present on some, okay? The same way that a cell wall is present on some eukaryotic cells, but not others, right? Plant cells have cell walls, but animal cells do not. Okay, so there are a certain contingency of cell walls, of bacteria that have cell walls, but they all have plasma membranes. And here you can see the ribosomes kind of floating around these little brown dots that we see here. This area in the middle, which you can see what looks like purple spaghetti, that is the nucleoid region. That's where the DNA is kind of kept. Now, there's not a whole lot of room in these bacterial cells because they're quite small. So there's the nucleoid region is pretty much right in the center. Okay, Another thing we see a lot of bacteria have are these flagella. Now, not all bacteria have flagella, but they're including it here, and that's a mode of transportation. And then we see these things up here called pili. We're not going to necessarily discuss what pili are, but pili are a type of way that bacteria can actually have sex, which is a really odd thing to think about bacteria having sex, because most of the time bacteria reproduce asexually. Okay, so a lot of the differences arise in kind of the external structure, as well as obviously the fact that there are no internal membrane-bound organelles for the bacteria, the prokaryotes. Okay, move on to the general characteristics of the eukaryotic cells. They have DNA in a nucleus that is bound by a nuclear envelope. Okay. Now we're starting to get into this idea of eukaryotic cells having these membrane-bound organelles, these membrane-bound compartments that allow them to do very specific things in very specific places and keep them there until they're needed. Okay. Eukaryotic cells are generally much larger than prokaryotic cells. I will probably, I've got a really good graphic that I probably should have included in here that shows the relative size of eukaryotic cells to prokaryotic cells to then viruses, and eukaryotic cells dwarf prokaryotes. Okay, they're very, very big compared to prokaryotic cells, and then when you look at the size of prokaryotic cells to viruses, those prokaryotic cells also dwarf the viruses. So we've got these varying levels of size in our microorganisms, but by far the eukaryotes are much, much bigger. Okay. So it says at the bottom bullet there, the logistics of carrying out cellular metabolism sets limits on the size of the cell. We are going to get into the size of the cell and how the size of the cell influences cellular metabolism, and also the cell's ability to get nutrients into itself and also kick out waste to the surrounding environment. That is something we're going to get more deeply associated with in Chapter 7. That's what our first lab is really based about. So to discuss this last bullet point idea, okay, let me get the pointer back out. To discuss this last bullet point idea right here, I think is a really good transition between Chapter 6 material and Chapter 7 material, okay, because it will be directly related to the cell's membrane, okay. So here's our idealized version of a eukaryotic cell. And it may look really crowded in there, and I say idealized for a reason, because it is just that. There are oftentimes many more Golgi apparati, or apparatuses, I'm not sure of the plurality there. There are many, many more mitochondria than just 3. There are many more peroxisomes than just 1. There's many more. There's much more surface area of the endoplasmic reticulum than what you're witnessing there. One thing I really do like about this picture is the fact that it does show how crowded things actually are. So one of the things, the misconceptions that students always have, and this is not going to really ever be tested on, at least by me, and I don't think the College Board, one of the misconceptions that students have is that the inside of the cell is extremely spaced out, and also that the cytoplasm is very liquidy. Both of those things are kind of incorrect. The inside of the cell is absolutely jam-packed with stuff, okay? It could be one thing or the other, but it's just there's stuff everywhere, okay? And it takes a while for things to move from one place to another, okay? And obviously on top of that, the cytoplasm is much thicker than what a lot of people originally think. Here's another example of a eukaryotic cell. Again, this is an idealized version. This is a plant cell, and you can see the difference here. We're going to get into some small differences between plant and animal cells once we start obviously talking about the chloroplasts and also the central vacuoles. Okay, those are the two big differences between plant cells and animal cells, on top of the fact that the plant cells have cell walls, which we can obviously see here. So we'll keep on moving. All right. In section 3 here, we're going to just briefly gloss over the nuclei, okay, the nuclear area of the cell. And again, this is not something that is new to you, I'm guessing. So I'm going to, I guess, not breeze through it, but I'm not going to spend a whole lot of time on it, okay? So the nucleus contains most of the cell's genes and usually the most conspicuous organelle in the thing. It's usually the biggest part. It often stains really well when you're looking at it under a microscope, and that's because it's full of DNA, which is extremely stainable. Okay, I don't know if that's a word, but DNA stains really well, as does the nuclear envelope, which is composed of very similar things to the cellular membrane. Okay, with a few additions, and I'll point some of those out here in a second. All right, so the nuclear envelope encloses the nucleus, separates it from the cytoplasm, and there is a distinct reason for that, and I'll kind of talk about it here. So we do have this. I'm going to bring up again a little snapshot picture of this so I can draw on it. Okay, so in the picture we see right here, we've got our nuclei. Okay, the nuclei is right here. The nucleus, I should say, is right here. It is surrounded by endoplasmic reticulum, which I'll spend a little bit more time on when we get into tomorrow's the next video. But one thing I want to mention here is the nucleolus. I don't recall this being part of the nuclei that the College Board really cares that you know about. But I always think it's kind of important to mention that the nucleolus is the place where ribosomes are created. Okay, so the nucleolus does have a function. It's generally, if you're staining this thing, it is the darker part of the nuclei, the nucleus, right in the middle there. Okay, and then on the outside we see the nuclear envelope. It's actually it's got an inner and outer membrane, so it's double layered in this case. It's got some area right in the middle of the It's almost like a fluid-filled matrix, and it's actually continuous with the endoplasmic reticulum, which we see here. Okay, so its little compartment is continuous with the endoplasmic reticulum. So the nuclear membrane kind of transitions into the endoplasmic reticulum. We also see the surface of the nuclear envelope has pores, okay, and the big difference here, the big function here of the nuclear envelope is, number one, to keep the DNA inside. Okay, it's a protectant against things that want to get into the nucleus, and it's also a protector for the DNA. Okay, so we want to keep this pristine copy, what we call the chromatin here. The chromatin is all of your DNA that's been basically unraveled. Okay, we still call them chromosomes, but when it's unraveled, we call it chromatin. And so we look inside there. We've got all this chromatin, this DNA. We want to keep these pristine copies of the DNA protected in one centralized location so that we can use them if we need to. Okay, so that's one of the reasons for this nuclear envelope. But we also want to be able to take the information that is in the nucleus, inside the DNA, and transport it out of these pores. Okay, so in here would be where the nucleus is, and we can transfer that information out through the pores. So what actually happens here, and we'll touch on this more in chapter 16 and 17, inside the nucleus we will actually make disposable copies of our DNA called mRNA, messenger RNA. Okay, and that messenger RNA actually leaves the inner part of the nucleus by way of the nuclear pores. Okay, so we get those little mRNA strands out of the nucleus, and once they're out of the nucleus, they combine with the ribosome and they make proteins. Okay, so we want to basically we want to segregate this really important DNA away from everything out inside of the cell that could possibly harm it, which is why we send that disposable copy of the DNA, which is the messenger RNA, out of the cell and into the general cellular environment. Okay. Now the nuclear lamina, as I mentioned before, helps to give. I didn't mention this before. I meant the nuclear envelope, as I mentioned before, is similar in construction to the cell membrane, but it has this stuff called nuclear lamina. Okay, and nuclear lamina is it's almost like rebar. It provides structural support to the nuclear envelope so it doesn't collapse in on itself. Okay, so enough about the nucleus. Talked about the general structures of the nucleus and what they're used for and why it's compartmentalized and things of that nature. All right, next up, ribosomes. Ribosomes, you guys again have heard of them. They are protein factories. We will talk again much more about ribosomes when we get to the chapter that deals specifically with proteins and creating proteins, because ribosomes are the protein constructors. Ribosomes help to create the proteins out of amino acids. Ribosomes are actually made up of particles of RNA. Okay, and that's another thing, again, when we get to chapter 17, that I really like to point out to you, is that when we see them in these diagrams and things like that, here's like a ribosome, a couple ribosomes, they just look like little brown balls a lot of times, or little brown chunky masses. But those things are actually composed of RNA molecules that are produced up here in the nucleolus. Okay, so they are composed of nucleic acids. They are nucleic acids of one variety or another. One thing we'll realize once we get further on in this material, again, this is not something you necessarily need to know for this particular chapter, but there are many different types of ribosome RNA that have many different types of functions. And one of the types of RNA is called ribosomal RNA, and its function is to make ribosomes. Okay? So ribosomes carry out protein synthesis in two locations. Sometimes they just float around in the cytoplasm. They're called free ribosomes, and they'll attach themselves to an mRNA molecule, and then they'll make some protein. You'll oftentimes also see ribosomes bound up to the endoplasmic reticulum or even the nuclear envelope. And these are called bound ribosomes, and these ribosomes actually will take mRNA directly from the nucleus as soon as it leaves the nucleus, the mRNA, and it will translate it almost immediately. So it's like trying to get this protein made as soon as possible, the ones that are bound up to the ER and the nuclear envelope, okay, because they are the closest to the source of the production of the mRNA. Okay, so in these two things, the mRNA and the ribosomes and the nucleus, I've provided a lot of detail that might not necessarily be needed. Maybe good to know. Okay, and it's good to start to know that stuff early on. But there's a lot there that I'm not going to say is needed or not on the test, mainly because I haven't produced the test yet. So, yeah. So where are we going from here? In the next video, I will speak relatively extensively on the endomembrane system. Okay, the endomembrane system, we're going to touch on the endoplasmic reticulum. We're going to talk about the Golgi apparatus in much more detail than maybe you've had before. Talk about the lysosomes also in a lot of detail. Touch on the vacuoles quite a bit as well. So we're going to pay more attention to some of these, I guess you'd call them ne'er-do-well ones, the ones that kind of get left out. The ones that always get the press are the ones like the mitochondria and the chloroplast and the nuclear and the nucleus, because they're kind of the big ones. They're the ones that we would consider really important. But I'm going to spend less time on those ones. I'm still going to talk about them, but we're going to spend more time on some of these ones that don't get all of the pub that the big dogs get. So that's what we're going to talk about in the next one, and I will get that done as soon as possible. Thanks, guys.